Hey everyone, welcome back to the channel. So today we are going to go over AQA AS Biology Paper 1 predicted paper for 2024. This is a walkthrough solution, but make sure before you get started, if you want to access the paper so that you can try it before, it will be on my Patreon link below. And also, if you find this content helpful, don't forget to subscribe for more exam tips and trips and other predicted papers for the rest of the exam season 2024. And just a quick reminder that while this is a prediction, it's very important for you to revise every topic that might come up because you never know what actual topics will come up in the real exam. So let's get started with question 1. Question 1. Some lipids. Describe how you could use the emulsion test to show that a seed contains lipid. So they already told us what type of tests, which makes it so much easier. This is the test 2 for lipids. So there's the same thing over and over again. It's always these steps. So first you want to crush. You crush the seeds and then you crush them with ethanol. This just allows it to break down so that we can actually, all the different components within it can actually separate. So the lipids can actually separate. Then after that, what you do is that you add water. This water is always added after ethanol. So you need to be written it that way where you add it after the ethanol. And if it is a positive test, it will find emulsions, which basically mean it will go white. And if that happens, there is lipid present. So B, a triglyceride is one type of lipid. The diagram shows the structure of a triglyceride molecule, which is over here. So a triglyceride molecule is formed by condensation. This is where two things come together, and when they form and they come together, they produce a water molecule. How many molecules in this triglyceride is formed? So we have the 4 different components over here. So it means 4. So 2i, the structure of phospholipid molecule is different from that of a triglyceride. Describe how a phospholipid is different. So if you see over here, this is a triglyceride. You have 3 fatty acids all connected to one glycerol. So a phospholipid is the exact same thing, except they do not have a fatty acid, one fatty acid. They only have 2, and this one's actually connected to a phosphate group. So that is the key difference between what a phospholipid is and what a triglyceride is. So use the diagram to explain what is meant by an unsaturated fatty acid. So an unsaturated fatty acid, just seeing the word unsaturated means that there is a double bond, and this is between 2 carbon atoms. So if you see over here, you can see that this over here is what we mean. So you can see that fatty acid C has an unsaturated because it has a double bond in comparison between 2 carbon molecules. So because they say to use the diagram, make sure you always refer to it, since one of the marks will always be referring to the diagram. Question 2. A triglyceride are taken into the body as a part of a balanced diet. These triglycerides contain fatty acid, include omega-3 fatty acid, and it has been discovered that these fatty acids are associated with health benefit. The benefit includes faster development of nerve cells and clearer vision. So it's also associated with protection from heart disease, arthritis, and cancer. The following figure shows how these are taken in and used by bodies of animals. So you can see that it's taken in, digestion, and then it's transported to the blood. This is pretty normal. And then what they're used for, they are used to make plasma membranes, respired for energy, and convert to other different fatty acids. So the question asks, use the information to figure to explain 2 ways in which fatty acids are important in the formation of a new cell. So this actually is already given to us by the diagram. We just need to make sure that we read it correctly. It already tells us how they are used. It's used for plasma membranes, respired energy, and fatty acids. So because they talk specifically about fatty acid, we can say that fatty acids are used to make phospholipids. And what phospholipids are used in, they are used to being membranes. So in the cell membrane, what these are made out of are phospholipids, which means more phospholipids means more membranes. So number 2, fatty acid can be respired to release energy, which we can see over here. It tells us, and this energy can be used for cell production. It can be to make energy, and this just means that more triglyceride means more energy is released. Question 3. A group of students carried out an investigation to find the water potential of a potato tissue. These students were given a potato and 50 centimeter 3 of 1 mole solution of sucrose. They used this solution of sucrose to make a series of different concentration. They cut and weighed a disc of potato tissue and left them in these solutions for a set time. So they then removed the disc and reweighed them. The table shows the process results. So explain why this data in the table above is described as process result. So process result just means that the calculations are already made from raw data and they only show what the calculations, like the value after the calculation has been made. They do not show raw data. So if you can see here, percentage stage of mass of tissue basically means that the raw data would have just been initial and final mass. And what we did is just that we minus them and we only showed the percentage change, the actual thing. We don't actually put in what the initial was, what the final, we just put what the calculated results are. So B, describe how you would use a 1 mole concentration solution of sucrose to produce 30 of 0.15 solution of sucrose. So what this is here is that you'd basically use 4.5 of the solution and you would use 25.5 of distilled water. Okay, so question C, explain the change in the mass potato in the 0.4 mole solution of sucrose. So if we go back up here, we need to explain this over here. So what we can see is that the percentage change of the mass is actually a negative. So what this means is that the water potential of the solution is less than the potato tissue. So it was more negative than the thing, which meant that the tissue loses water, and every time we say that it loses water or a specific substance, we need to say how it's transported, and this is transported by osmosis, and that's why it has a negative. So D, describe how you could use the student's result in the table above to find the water potential of a potato tissue. So what you do is that you basically plot the graph. You'll have concentration on the x-axis and you'll have percentage change in mass. So then once you plot your graph, kind of looks something like that. And wherever it crosses the x-axis, which basically means when the percentage change is 0, you find that concentration over here. And then what you do is that you use different resources, and then that's where you can find the water potential of the sucrose concentration. And then that's how you get the answer. With these type of questions where they always ask, it's always the same 3 points that you have to add. So you just put these every single time. Question 4, the figure below summarizes the process of meiosis. The circle represents cells and structures within the cell of the chromosome. So describe the appearance of one of the chromosomes in cell X. So you can see here, these are 2 chromosomes, but we only need 1. So let's say we'll circle this one. So it says describe. Describe just means we say what we see, but because it also says explain over here, we need to say why does it look like that? Especially since it's 3 marks, we know we're going to get 1 mark at least from explaining. So you can see over here, there is 2 chromatids, and these are joined together with this dot over here, and that is called a centromere. So that already here is 2 marks, and why this happens is that usually it's just 1, as you can see over here. However, because DNA replicated, because they're going over this with interphase, that's what happens. That's why that second one over here came. So the explain part is because DNA replication has occurred. So B, describe what has happened during division 1 in the figure above. So we can see here it's division 1. So what you can see is that each one goes away to different ones. So chromosomes in a homologous pair, which is these homologous pair, mean that they are just the same, they are paired together. So, and then each one of them goes to its own daughter cell, and this is a haploid number. So C, identify the event that has occurred in cell division 2, but not cell division 1. So you can see over here. One thing that you can say is that the centromere it's got the division of the centromere, so each one has its own. It's no longer connecting to. And also because it's no longer connecting to, you can say that there was the separation of the sister chromatid sister, because they're basically the exact same. They're just a replicated copy. So D, name 2 ways in which meiosis produce a genetic variation. This is always the same 2 over and over again, so make sure that you learn it. And it comes up pretty often in this question. This is because of independent segregation, which is the same as random assortment, and also because of crossing over. And this only happens in meiosis. That's why we get that we are so different from, let's say, our sisters, from our parents. These type of things is what makes every single one of us are so different, and there is variation in our genetics. Question 5. Micronesia is a group of islands in Pacific Ocean. So the white-fronted ground dove is a bird found in these islands. So the diagram below shows how these are classified. A, to which class does the white-fronted dove belong to? So if you go over it, we have domain, kingdom, phylum, class, order, family, genus, and species. So you just need to remember there is lots of different mnemonics that you can use so that you can memorize this. So one of them is dear King Philip came over for good soup. So if we see over here, we can see that this is the kingdom, since it's Amelia. Then we have that is the phylum, and this over here is the class, so it's apes. So give the scientific name for the white dove. The scientific name, this is always the genus and species. Those are the only things that we count in a name, so it would just be this over here and this. C, the classification system consists of a hierarchy, as there is small groups within larger classification. Give one other feature of hierarchy shown in the diagram. It's always the same, it's always this, and the other feature is that there is no overlaps. So if you see over here, it is not like a Venn diagram where there is 2 things that overlap. Every single one is separate from another. However, there is something that's bigger and something that's smaller, which is this hierarchy. So question 6, describe and explain how a countercurrent system leads to efficient gas exchange across the gills of fish and water. So what it is that, let's say this over here is the fish and this over here is the water. So while the fish, their blood goes this way, while water goes this way. This means that because they flow in opposite direction, there is a concentration gradient always. So while it's high over here, it's low over here. And where it's low over here, it's high over here, which means that there's always this concentration gradient where they're able to go and they're able to exchange resources that they need. Let's say in comparison, if they were both going the same direction, like over here, they're both going the same direction, this will both be high concentration in both ways, and there will be still low concentration both ways, which means after a while, their concentrations will be the exact same, so there's no more movement and gas exchange between them, which of course is inefficient because they want to get as much resources as possible. So them having different concentration at different time is the reason why gas exchange is so efficient. And this happens across the whole gill, not just parts of it. So B. Amoebic gill disease is caused by a parasite that lives in gills of some species of fish. So the disease causes the lamellae to become thicker, this is important, and to fuse together. So it reduces efficiency on gas exchange, and fish. Give 2 reasons why. So while they're talking about fish, this is basically the same. They have the same efficiency, same reasons why they're so efficient as a human does, as any living organism does, and this is when it has larger surface area, and when it's thinner, we have more efficient. That's the same with our alveoli. So we just apply the exact same principle. So it says over here that becomes thicker, so this means there's a longer diffusion distance, which of course makes it less efficient. And it also says that it fuses together. Because it fuses together, that means there's reduced surface area. So always make sure that you remember these 2 reasons and try to use it in the application that they gave you. These type of questions just wants to test how you apply your already given knowledge into a question. So even if you see a question that you completely don't understand, you do know the answer, you just need to find a way to know your existing knowledge into what the question really asks you to do. So C, the table below shows some feature of gas exchange of a fish at rest. So calculate the volume that would have to pass over gills each hour to supply the oxygen required by fish. So show your working. So what this is that you basically do 0.4 times 90, you do the oxygen required for fish per kilogram. So that means we need to times it by the mass. This will equal 36. And this is the volume absorbed of each water centimeters. So that means you divide it by 7, and which is 5.14, which is the answer. Always try to put it at least into 3 significant figures, 2 decimal point places. So don't just round it to the whole number. So the volume of water passing over the gill increases if the temperature of the water increases. So suggest why. This is mainly due to increased metabolism. When the temperature increases, there's increased metabolism, increased respiration, also increased enzyme activity. This is generally because of increased kinetic energy within the water. So question 7. Name the process by which oxygen passes from the alveolus in the lungs into the blood. This is just called simple diffusion. Describe 2 adaptation of the structure of alveoli for fish and gas exchange. So like the one that we said before, it's always the same 2, and this is thin walls and also that the surface area is large. This is because there's lots of alveoli, and because there's lots of this fold, there is more place and more surface for things to be able to go in and out and for the reactions to occur. B, the photograph shows a fire-breathing bull catching fire. So fire breathers do this by blowing fine mist of paraffin onto a flame. So some mist can be inhaled and eventually leads to fibrosis. So people who have been fire breathers for many years often find that they cannot breathe properly. Explain why. So this is just on the knowledge that you know what fibrosis is, and what that is, it basically it lets the lungs lose electricity, and because of this they're unable to recoil, which means that there is decrease of surface area, so they're unable to be as efficient as normal when it comes to gas exchange. Question 8. Ecologists investigated the size of an insect population on a small island. They used a mark, release, capture method, which is basically exactly what they do. They mark it, release it, and then they recapture it to try and find out what the population is. So to mark the insects with a fluorescent powder, this powder glows bright red when exposed to ultraviolet light. A, the ecologists captured insects from a number of sites of the island. Suggest how they decide where to take their samples. So it's always the same with these type of questions. It's a really nice topic because the answers are always repeated. So what you do is that, let's say if this is the area that we want, you would draw a grid so that you kind of split it into sections, and then what you do is that you can name it like that. You can name this 1, 2, 3, 4, 5. And at random, the most important part is that it's random so that it's not biased and there's no less likely of anomaly happening. You take a coordinate, so let's say B2, and that will take another one, it will be like D5. You'll take that sample over there. So B, give 2 assumptions made when using the mark, release, capture method. There is a lot of assumptions that can be made over here, and it's always the same no matter what, so I do really recommend that you learn at least 2. I like to use no immigration and birth rate is equal to death rate since it's the easiest to remember, but the other ones that you can have is that no losses to predation, marking does not affect the survival, and in this case they all belong into one population. So C, suggest the advantage of using fluorescent powder in this experiment. So what it is, it says over here that it only glows bright red when exposed to ultraviolet light. The fact that it only glows brightly with UV is that it makes the insects not more visible to in their day-to-day life. So if they were visible, it means that they were more likely to be caught, meaning they're more likely to increase predation because of it. So because this doesn't affect their survival rate, it means that we are sure that there is nothing, that we didn't harm them in terms of ethics, and also that the population is more accurate. So the ecologists did not release the insect after they captured 1 to 5 in the release of marked, so not. So calculate the number of insects in 1 day after released marked insect. So how you do this is that you do what was marked in the beginning, you times this by the amount that was captured, and you divide it by the recaptured one. This is the equation that you always use. So we times this together, and if we time them together, we should get, you get 790,192. You divide this by 78, and the answer is 10,130. So the ecologists expected to obtain the same results from their calculation of the number of insects of this island on each day during period 1 to 5 after release. They estimated the number increased after day 1. During the same period, the number of insects they caught decreased. The method used by ecologists might have caused these changes. So suggest one way in which the method used by the ecologists must have caused increase in the estimate size of the insect population. So already the fact that they did not release it is already such a big red flag. So we know that that is the reason why. So it's because scientists removed large numbers of insects, and because they did not return them to the same area and the same population, this affected the ratio of marked to unmarked. We know it's division. So a bigger or smaller one number is because of it is going to change what the overall calculation is. So question 9, a mutation in a tumor suppressor gene can form a formation of a tumor. Explain how. So this specific gene suppresses the ability for tumor. But with this mutation, this means that this mutation can either inactivate it, so this gene completely doesn't work. It could also make it not even in control, or it could slow it down. So there's lots of different ways that can happen. So because of this, the rate of cell division is going to happen really, really fast and out of control, since that gene is no longer able to suppress to control the cell division rate. So 2I, not all mutation result in a change of amino acid sequences encoded in polypeptide. Explain why. There's always 2 reasons why. One is because code is degenerate. This basically means that there is more than one code for an amino acid. More than a triple codon can code for the same amino acid. So let's say UGG and UCG both code for the exact same amino acid. So even though this one could have changed, they would still code for the same amino acid, meaning tertiary structure is still the same, meaning the protein function and shape is still the same. What also can happen is that there could have been a mutation in the intron, and we all know that mutation in introns are cut out. This is pre-mRNA, they cut out before they go on to translation, so nothing would affect it. A mutation in the exons, however, could affect. So B, some cancer cells have a receptor cell in their cell surface membrane that binds to a hormone called a growth factor. This stimulates cancer cell to divide. So they have produced a monoclonal antibody that stops the stimulation. From your knowledge, suggest how this antibody stops the growth of tumor. So as soon as you see antibodies and monoclonal, we kind of already know that we're talking about protein, so it's the same kind of structure. So it's basically antibody has a specific tertiary structure, which means it has a specific binding site. So this is only complementary to the receptor protein, so it only binds to the receptor of that GF. So this means that it prevents the GF from the growth factor, which is from stopping it from binding. Okay, so question 10. The diagram shows the structure of the cell surface membrane of a cell. Name A and B. So this is the phospholipids I was talking about from before. There is 2 fatty acids, and this is the phosphate group over here. So we can see that A over here, this is the phospholipids. And B, this is one of the proteins that are embedded to help with transport of substances. This is a channel protein. Protein over here is a must. You must write that. Everything embedded is a protein. You won't get the mark unless you put protein, because that's what the main thing is. So BI, C is a protein with a carbohydrate attached to it. The carbohydrate is formed by joining monosaccharides together. Name the type of reaction that joins monosaccharide together. As soon as you see join, we know that this is a condensation reaction. If they said separated, this is a hydrolysis reaction. So a condensation basically means that they join together and then H2O. Water is also released. So 2I, some cells lining in the bronchi of lungs secrete large amount of mucus, and mucus contain protein. So name one organelle that you'll find in large numbers of a mucus secreting in the production. So one thing that you can find is a rough endoplasmic reticulum or even ribosomes. This is already known because we saw that it has protein. So the fact that it has protein means that it needs somewhere to be able to package it and to make the polypeptide, and that is the role. It's to make the protein and to form the peptide bonds so that they can actually be used to cell. You could also say a Golgi apparatus because that is able to package and process protein. Mitochondria, this is to release a lot of energy and make ATP so that this can occur. And also vesicles, this is so that it can actually transport. So now for the comprehension part of the papers. So read the following passage. So you see the herpes simplex viruses infect nerve cells in the face, which includes some near the lips. So like many other viruses, this can remain inactive inside the body for years. And once it becomes active, it causes cold sores around the mouth. So human cells infected with a virus may undergo programmed cell death. So while HDS is inside the body, only one of its genes is transcribed. This gene is the LAT gene that prevents programmed cell of an infected nerve cell. So scientists have found that transcription of this gene produces microRNA. The microRNA binds to some of the nerve cells of these own mRNA molecules, and the mRNA molecules are involved in the programmed cell death of the nerve cells. The scientists concluded the production of microRNA allows the HVS to remain in the body for years. It's very important that you read the passage that they give you in lots of details, because most of the time you're going to find that the questions already have the answers within the passage, or it will at least give you hints of what direction they want you to answer the question in. So use the information and your own knowledge. So HVS nerve cell in the face. So explain why it only affects the nerve cell. This again is because the outside of the virus has antigens and proteins, and with complementary shapes to the receptors and proteins in the membranes of the cell. And these are only found on the membranes of nerve cell. That's why it only affects nerve cells. So B, HSV can be remained inactive inside the body for years. So explain why this virus can be described as inactive. So this is because there is no more cell that's infected while it's inside of you. It's not really affecting anything. And why it's not affecting anything is because the virus is not replicating. Virus not replicating, it stays in one place. Once it replicates, then it starts to infect all these other nerve cells, and that's when the symptoms shows. So C, suggest one advantage of a programmed cell death. This basically just prevents replication of viruses, which is good because that means that it can be unaffected and you won't get the cold sores that they described. So the scientists concluded that the production of this microRNA allows HSV to remain in the body for years. So explain how microRNA allows it to remain in the body for years. And this is because microRNA binds to the cell mRNA, which they said in the passage. So this allows us to hint towards what they want, which is that they wanted to say that it binds by specific base pairing. Always put as much detail as possible on how things actually happen and do it in chronological order. So it binds to a specific base pairing. It also prevents mRNA from being read by ribosomes because it's already paired to that. So that means it's not able to have translation, as it wasn't been able to read. Because it prevents translation, it causes cell death. So that's the end of it. Please subscribe for more content. We will be doing chemistry, math, GCSE, and we'll also be doing A-Level biology. predicted papers. We will also be going over lots of different types of exam questions that are possible. That is the future plan. So comment down below if you have any questions. We'll be happy to help. And you can get this paper in the link below. Thank you so much.